Hi students, good morning to all. Today I am going to discuss about photosynthesis. So it is one of the important topic in plant physiology and easy to score good marks in that. Okay, so before entering into the topic, and let's have the refresh points. How do the living organism get energy? Obviously, we say the food provide energy. So where do we get this food? The prime source of food is a plant. How do we call the plant is a prime source of the food? Because these green plants prepare their own food materials just using the carbon dioxide, water molecule and sunlight. Okay, with the help of the carbon dioxide, with the help of the water molecules, with the help of the sunlight, the green plant is able to prepare its own food. The prepared food is 90% utilized by the plant, remaining 10% is stored in the plant's part. It may be stored in the leaf, or it may be stored in the stem, or it may be stored in the root. Okay, it may be stored in the fruit also. Okay, this 10% stored food materials only passed from the one organism to another organism. That follows the energy flow, 10% law. Okay, the energy from one organism to another organism is transferred only at 10%. Okay, the plant uses 1% of sunlight and prepared 100% of food materials. It uses only 1% sunlight and make it 100%. Okay, from the plants, then it goes on to the herbivores, then it goes to the carnivores, then it goes to the omnivores. It goes on like that. So when the food, this is we call it a food chain. In food chain, the energy flow, the transfer of energy from one tropic level to another tropic level is only 10%. This is we call it a 10% law. Okay, so the plant uses the carbon dioxide, water molecule, sunlight. All these things are the light energy, and this light energy, okay, and this with the help of the light, the plant is able to prepare this food. So light energy is converted into chemical energy. It is a biochemical reaction. It is, we call it a biochemical reaction. How this energy, this light energy is converted into chemical energy, that is the surprise of this topic. Okay, so we might have conducted in the previous classes, in our early classes, okay, 8th standard, 9th standard or 10th standard, how to test the starch in the leaf. What you might have used? You might have used iodine solution to test the starch and the plant leaf. Okay, so you might have exposed the plant to the sunlight of 12 to 24 hours. Then afterwards we might have picked that single leaf from the plant. Then it will be kept in the boilers, and along with that, so I mean it is kept in the test tube. Okay, that this test tube is filled with ethanols and kept in the boiler, the water boilers. Okay, so what happens when we boil it, automatically the green pigment which is present in the leaf will be removed. Okay, so for that removal only we are using the ethanol. So ethanol remove this green pigment. Afterwards we will take out and keep it in the watch glass, add a drop of iodine solution. If the starch is present, and it will show the blue black colors. So the blue black color indicates the presence of starch. Okay, then followed by that, in order to the plant need carbon dioxides. Okay, so what we have done, we kept that one plant inside the dark room, another is an outside, and this one plant, this plant, one of the leaf is covered with KOH solutions. Okay, so another leaf is just exposed out. So this KOH solution absorb the carbon dioxide. Absorb the carbon dioxide. Okay. So this will prove that the carbon dioxide is necessary for the photosynthesis process. So when the KOH absorb the carbon dioxide, automatically that the leaf of that plants will not get the sufficient carbon dioxide to prepare the food materials. So it can't show the starch presence, and that which is exposed to the outside and which is not covered with the solutions, and this leaf will show the presence of starch, so that indicates how much the carbon dioxide playing vital roles in the process of photosynthesis. Okay, so let's discuss about the earlier experiments. So today we know that the photosynthesis reactions. Okay, then that at the end of the photosynthesis, at the product of photosynthesis, we call it an carbohydrate, we call it an water molecule, along with that oxygens. How it is possible? That is only we are going to discuss. So in the earlier experiments, so the scientist who is called Joseph Priestley, okay, so he discovered the oxygens in 1770. So the discovery of oxygen is done by Joseph Priestley. Okay, fine. What he has done? He has done a sequence of experiments. Okay, in his first experiments, he has taken the live mouse and the candles. Okay, then the closed bell jar. Clear? So he kept the live mouse as well as he lightened the candles and closed with the bell jars. Okay, closed with bell jars. He kept this mouse as well as the candles under the closed bell jars. What happens? After some times, the mouse dies as well as the candle stop burning. The candle stop burning. So from that, what he has concluded, okay, so that the air is damaged, the air quality, the nature of the air is damaged by the mouse as well as the burning candle. The mouse as well as the burning candle. So the burning candle and the live mouse is died because of the air is damaged by them. So he wanted to conduct the second experiment. So what he did, he kept the mint plant along with the live mouse and the burning candle and closed these things with the closed bell jars. So what happens? And this mouse able to survive and the candle able to continue burning. Okay, so from this what he has concluded, the damaged air is restored by the plant. So in the first experiments, we only kept the live mouse and the burning candle under the closed bell jar. So what happened? The candle after some times then extinguished and it stopped burning. The same way the mouse got suffocated and died of that. Okay. So but in the second experiments, when he kept the mint plants along with the live mouse as well as the burning candles, then he was able to see that the mouse is able to survive and this candle continued its burning. Okay, from that what he has concluded, the plant restored the damaged air by breathing animal as well as burning candle. The plant restored the damaged air by breathing animals and burning candles. Okay, in order to prove this, and another persons, okay, John Inkins, and he is the person, and is what he did, no, he kept these things in the dark and light. It's an elegant, he has done the elegant experiments with aquatic plants. So we kept aquatic plants inside and dark under light. Okay, in light exposures, the light regions, what happens? That small droplets, air bubbles are identified around the leaves. When this aquatic plant is exposed to the light, so around this aquatic plant, and small air bubbles are identified, but it is absent in the dark. I mean, it is absent in the plant which is kept under the dark conditions. So thereby, in the light conditions, when these aquatic plants are kept and it is producing the air bubbles, and he finally proved that this is the air bubbles of oxygen. This is only oxygen. So he proved that the plant produce oxygen, okay, and it consume the carbon dioxide. So it intake the carbon dioxide, evolve the oxygens. So in 1854, and the person's called Van Sach, okay, Julius Van Sach, okay. In 1854, he proposed the concept: the plant produce glucose. The plant produce glucose. He told the plant will produce glucose. As soon as the glucose is produced, it is converted into starch material. So usually the glucose, as soon as it is produced, it will be converted into starch materials. He also told that the green substance present in the plant is especially present in the special bodies within the plant cell. That special body is nothing but the chloroplast. The special body is nothing but the chloroplast, and the green pigment is nothing but the chlorophyll. Okay. So he has proved only the plant is able to produce the glucose. Okay. So during the photosynthesis, as the plant grows, then the glucose also produced by the plant, and as soon as it is produced, this glucose is converted into a starch. Okay. And also he has proved that the green substances presence in the special bodies, the special bodies later as we call it in chloroplast. Okay. So in 1854 the experiment was like that. Okay. Followed by him T.W. Engelmann. So this T.W. Engelmann has proposed the theory, and he told which part of the spectrum is able to absorb the light. So in the previous experiments we were explaining the light is the source, energy source for the plants to prepare its food, and this T.W. Engelmann wanted to know which spectrum of the light is preferable by the photosynthetic plant. So in order to find out these solutions, he has conducted the experiments. So he has taken the suspension of aerobic bacteria, which is illuminated with cladophora as well as then green algae. Cladophora as well as green algae. He has taken a prism. So he passed the light on the prism. Prism split the light spectrum into different spectra. Okay, and it is exposed to the suspension. Already I told this suspension contains that aerobic bacteria illuminated with that green algae and cladophora. So when he passed the light on the prism, the prism split out the light. Okay, make it many spectrum of the light regions. Okay, so certain spectra which are observed by this green algae and cladophora. Okay, that spectra is nothing but a blue and red light. Blue light as well as red light spectrums are the favorable for the photosynthesis activity. How? So where the blue light and red spectrum is exposed on the suspension, around that he was able to identify the numerous aerobic bacteria. The aerobic bacteria comes around the region of the red light spectrum as well as blue light spectrum and suspensions. So he concluded that the The favorable spectrum of the light is blue light as well as red light, which is unfavorable for the photosynthesis activity. Photosynthesis activity. Okay, fine. By 19th century, in the middle of the 19th century, and the scientists are able to proclaim, are able to say that the photosynthesis is a chemical reactions. Okay, and this plants uses light energy sources and to make it food materials, to make carbohydrates. Okay, so with the help of the plant uses light energy with the help of the carbon dioxide and water molecules and able to prepare the carbohydrates, and with the evolutions of the oxygens. The empirical formula of this reactions is CO2 plus H2O in the presence of light, and we are able to get carbohydrates, CH2O plus O2. The CH2O is the empirical formula of carbohydrate. It may be the glucose, it may be the sucrose, okay, whatever it is, and it is a carbohydrate. Clear? And the 6 carbon atom molecules. 6 carbon atom molecules. Clear? And in Cornelius Van Neel. So Cornelius Van Neel, he demonstrated that the plant essentially needed of light. Okay, so Cornelius Van Neel demonstrated the plant essentially depend on the light energy. Okay, to get an hydrogen donors from the suitable oxidizable compounds and reduce the carbon dioxide into carbohydrate. So Cornelius Van Neel, he was demonstrating that the plant essentially is in light dependent reactions, where the hydrogen donor from the oxidizable compounds reduces the carbon dioxide into carbohydrates with the evolution of the oxygens molecules. Okay, so look at these equations. So here 2HTA and carbon dioxide provides 2A and CH2O plus H2O. How we was able to demonstrate that plants need essentially the light, and which is converting, so the plant photosynthesis reaction is a light dependent reactions where the hydrogen is donated from the suitable oxidizable compounds, reduces the carbon dioxide into carbohydrate. How he was able to explain his concept? So he conducted, in order to prove that, he conducted the experiments by using a purple as well as the green sulfur bacteria. Purple and green sulfur bacteria. So all the organisms will not use the hydrogen donors from the water molecules. Okay, example, the purple and green sulfur bacteria, instead of hydrogen molecules and H2S, hydrogen sulfide acts as an hydrogen donor. Okay, in case of purple and green sulfur bacteria, instead of water molecules and this hydrogen sulfide H2S acts as a hydrogen donor. So from that hydrogen is donated and it forms then hydrogen sulfides or sulfate in the end of the donations. Okay, so thereby this Cornelius Van Neel concluded this hydrogen donor is not coming from the carbon dioxide. It is not from the carbon dioxide. It is solely from the water molecules. It is solely from the water molecules. So thereby this hydrogen donors, as he explained, this photosynthesis is a light dependent reactions where the hydrogen donors are from the oxidizable compounds, reduce the carbon dioxide into carbohydrates. So the hydrogen donor solely comes from the water molecules. Solely comes from the water molecule. It is not from the carbon dioxide. It is not any other sources. It is not from any other sources. The source of water molecule, source of hydrogen is solely from the water molecules, not from the carbon dioxide. Okay, not from the any other substances. Clear? And it is completely, the photosynthesis is completely a light dependent reactions. Okay, so thereby he has brought the equation 2H2A plus CO2 gives 2A plus CH2O and water molecules. That can be written as an photosynthesis reactions, the letters as CO2 plus H2O light energy gives C6H12O6 plus H2O with the evolutions of water molecules. The evolutions of water molecules. This is the photosynthesis reactions. Okay. Photosynthesis reactions. So here equation is not balanced. 12 we are adding, and here also we are adding 6. Okay, and here got it clear. And this one we will be having 6. So now the equation is balanced. So 6CO2 plus 12H2O gives glucose molecule, that is C6H12O6 along with the water molecules and oxygens. Okay, 6H2O plus 6CO2. So thereby we are very clear how this hydrogen donors come from and how this light is, the spectrum of light is used. Okay, which spectrum of light is used by the photosynthetic plants, and how this glucose is produced. And our doubt is now very clear. First things, plant alone prepared food materials. Okay, so thereby the green plants are known as an autotrophic plants. Means autotrophs. Okay, and for all the food preparations, for all the food resources, and the plant is the basic principles of everything. Okay, next thing is the plant uses carbon dioxide and eliminate the oxygens. Okay, so got it. The plant produces glucose and stored as starch materials. Do you understand? Okay, these green substances present in the special bodies within the plant cell. That special body is nothing but that chloroplast. The carbon dioxide and water molecules are converted into glucose molecules with the evolution of the oxygens. Okay, so thereby we can conclude the carbon dioxide is reduced into carbohydrate. The water molecules act as an hydrogen donors to prepare the carbohydrates. Okay, do you understand? And enter the photosynthesis process, and we get the product of glucose as well as water molecules as well as the oxygen molecules. So all these things are proved by the many experiments. Got it? Clear? Thank you so much for watching my channels, and in the next class we will see where does the photosynthesis take place. Where does the photosynthesis take place?